Thank you so much, Deepa, for agreeing to do this. And for anybody who's not familiar, this Ask Me Anything session with Dr. Deepa Agashe is a part of our initiative. I thought, sorry, I thought there was some feedback. So yes, this is a part of our initiative called Lab in Focus. And it's a chance for us to take you behind the scenes of a lab and have the lab tell their own stories in their own words. And we're very glad that Deepa's lab has participated so enthusiastically. There's been a Instagram takeover. There have been a lot of stories on Instagram, as well as papers shared on Twitter and all of that. So I'd encourage you to check all of that out, and we'll share the link. But for now, what we want to do is really hear from Deepa and see what her answers are to all of your questions. So without any more introduction, I'm just going to hand it over to Deepa, and I'm assuming all of you know that she runs the adaptation lab at NCBS. Deepa, maybe you can start by telling us how you joined NCBS, since you're celebrating 9 years here.
Yeah. So I joined as a faculty here in 2012, and before that, I was doing a postdoctoral fellowship in Harvard in Boston, and it was really fantastic to start my own lab. And I was so excited because there were so many questions in my head that I wanted to answer, and only two hands and only one brain that could think. So it was wonderful to have the chance to start working with many people and together address all of these different questions. And along the way, of course, we added many new questions, many more people joined. And as people joined, they started asking their own questions, or they found interesting twists to the questions that we had in mind to begin with. We found something interesting, and that led us, in unexpected directions. People joined the lab who were coming in with their own different ideas, and so it's been a very very fun journey, and I look forward to many more years of this journey continuing. Um, yeah, I guess that's kind of how we started. It was a bit slow when we started because it takes time to find the right set of people. It takes time for people to catch up to the concepts that the lab would require, to catch up to figuring out how to function, how to work in this environment, which was new for all of us. And when I set up the lab, we were one of the first labs to also set up in a new building at NCPS. So the building was not set up, everything was basically this giant empty floor. And so we were very, very fortunate to have a couple other colleagues lab nearby, who were already set up and who used to, who did bacterial work in Ashwin's lab. And so we borrowed and stole a lot of things from them. And it was really nice to have that help starting on in the early days. And I would say I was also very lucky that at that point, a bunch of the first few students who joined the lab were really very enthusiastic and were incredible in setting up the lab, because they were the ones who were, after all, working at the bench. And so it was really fun and really nice that they were very good natured about it. They had to set up everything because nothing existed at the point. So that was the start.
Yeah, that's great to hear. I mean, a lot of people don't think about the infrastructure of a lab. So that's good to hear that, you got your students involved in actually physically setting up the lab too.
Yeah.
Yeah. And the so I just wanted to add, for anybody who's kind of curious about anything and they're watching on YouTube, please put your questions in the chat there, and obviously on Zoom the Q&A box is there, or you can unmute yourself, but please raise your hand before you do that, right? So we know that you have a question. And right now we have one question already. So when you were applying to NCBS, what's the kind of process that went into that?
You mean the application itself?
Yeah, so the entire question is, can you describe your journey towards becoming a young investigator and applying to NCPS? So what's the difference between a young investigator and an assistant professor at NCPS?
Yeah, so NCBS has this different non-tenure track program called the Young Investigator Program, and that is meant for people with relatively less postdoctoral experience, or maybe in some cases no postdoctoral experience. And the point is that you might want to you might sort of be ready already to start your own lab, but you don't have the extensive experience that a postdoctoral or long postdoctoral stint will bring. And so the program is meant to get things off and running for somebody like that, especially if they want to start something different, something new. Not continuing on their trajectory which they had followed in their PhD and postdoc career, which is sort of usually what people do for their own labs when they set them up. So this was a nice chance and opportunity to set up my own lab without sort of having, a large experience and a large sort of set of publications or a lot of work accomplished in any one field. Because what I had also done is I had switched fields dramatically between my PhD and my postdoc. So my PhD work, I worked with a lot of sort of standard organismic biology, working on ecology and evolutionary dynamics of adaptation with flower beetles. And I did not do any genetic work at all during all of this time. Whereas for my postdoctoral work, I started working with bacteria, trying to ask how codon bias at the level of specific proteins is important for determining their adaptation or their fitness. And so this work required me to learn a lot of new molecular techniques. For me, new. I mean, they were standard techniques, but I didn't know them. I didn't have any experience with them. So this meant that, in the roughly 3 year postdoc that I did, I didn't get a chance to sort of make a huge amount of progress in that project that I built up. I also built up the project largely, completely new project for me, of course, with the help of my postdoc advisor. And so I guess, but I felt ready to start a lab. So I feel like under slightly different circumstances, I might have stayed on for a little bit longer, done more work, had more under my belt to show for my postdoctoral time, which I didn't at that point. But I wanted to start a lab. And so this was a really nice opportunity. I interviewed at NCBS. I really liked it. And so that was kind of what it was. And so the normal assistant professor, associate professor is what's called tenure track. And we have that. That is most of the faculty that you see at the moment are from that track. And some 3 years ago or so, I could apply to switch to that track. So now I am in the tenure track. I'm tenured. I'm an assistant professor here. And then at some point, I will get a promotion to associate and so on. So now I'm on that normal track. And so if any of you are interested in this Young Investigator program, I'll be happy to chat with you more. You can email me. I can talk to you with more details about the program, about the track, and what benefits or costs it might bring. It's not all great. There are some costs involved, but it depends on each person's situation, what would work best for them.
Okay, so great. So I think there's another question about, what you just discussed a little bit, right? So how does funding work, and what is it like? What is the kind of criteria that a funding agency looks at before they might provide a grant to you, especially maybe if you're starting out or if you're an early career researcher?
Um, I think there's actually, it's hard to have a single answer for this because our government funding schemes, particularly in India, there are so many of them and they're all very, very diverse. So, there are many different streams and tracks for which you can apply for funding. In general, I would say any funding agency across the world is looking for good ideas, right? And clear evidence that you've thought about those ideas, you have some backup in case stuff doesn't work out very well, and you have the intellectual and the logistical capability to address those problems that you're proposing. That's kind of what any funding agency across the world will look for. Now, exactly how much other factors play a role in whether you get a grant or not, that varies depending on the funding agency, the specific kind of funding it is. So, for example, you might have some funding which is earmarked for early career people, right? So in that case, they're not going to be looking for, okay, 50 papers that you should have published, whereas somebody who is applying as a full professor would be expected to show, a large body of work before they will, they will be trusted with more funding and so on. So these kinds of things will vary. There are in India, in the government agencies, there are many times very specific calls for funding, for a specific focal research area. So, for example, there was recently a call for chemical ecology involving the northeast, on northeastern states and regions of India. And the goal is to explore the biodiversity that's there in light of chemical ecology. And so they screened only proposals that, had this kind of work as their focus. So there are similar calls that come for specific areas. There are regular funding cycles, which sort of are regular DBT or DST or ICMR research projects which get funded. In a nutshell, that's kind of what funding agencies typically look for. I think this question also is asking about why molecular biology gets preference over ecological research in terms of funding. Um, I think actually I'm not entirely sure that that is even true. I think there are a lot of funding sources for ecological research as well, perhaps not from our traditional government funding bodies. But my impression is that there are actually a lot of philanthropic and other funding agencies which do provide a lot of ecological research funding in India at least. Um, it's true, I think, overall that government funding agencies do provide typically much more support for molecular biology work. Um, but I feel like that's changing. So, for example, DFT now has a panel dedicated to organismic and evolutionary biology and ecology, which is great to have, which didn't exist until very recently. So I do think there are many more, active sort of core ecology evolution scientists in India now, and hopefully the situation will change for the better in the future, where um, you know the idea that these are valid areas of inquiry and important areas of inquiry is sort of more taken more seriously. So we'll see how it goes. But what I would suggest is this is a question that I think is probably better answered by people who've uh, been in India for longer, slightly older professors who will have, I think, a broader view of this situation. So if you get a chance, I would definitely suggest asking them about this a bit more. And when I meet them next, I will ask as well. I'd like to know the answer too.
Okay, sounds good. I think, the next question is about how much time do you really get to spend doing experiments or, at your workbench because of the other responsibilities of running a lab and supporting your students and everything.
Mhm. So I don't get to do many experiments at all, but some of that I don't really miss very much. So I know some people do miss being at the bench and doing pipetting. I'm not particularly in love with pipetting. I'm fine without doing it. Um, what I would hate to miss is the planning and the design of experiments and the analysis. That stuff I really enjoy. So with my students, we really spend hours and hours talking about any piece of work that we're doing, any project. We also spend a lot of time on the analysis. So analysis is something that I do end up doing quite a fair bit of as well, and it varies from project to project. So that I do enjoy, and I've been able to do at least occasionally some work in with the flower beetles in the lab, and that is a little bit more manageable because the experiments are sort of large, but we often need focus, lots of hands on deck once in a while when there are very large experiments. So those are the times that I am able to chip in, and I sort of, it's a limited time commitment where I go, we work, and then I can leave and the others carry on. But that doesn't happen as much with the bacterial work that we do. And so, because that usually requires a longer term commitment, stronger schedule. And like I said, I don't then necessarily miss that very much in the work itself. Um, for the field work component that we did a little bit of, some of the projects that we did, I was able to go and do some collections for some of the field work. So I'm quite happy that I was able to do. But like I said, I think it works out really well for me. I enjoy the the thinking part of stuff and the analysis and the writing a lot. Um, and it's amazing when data appears in front of me, and I didn't have to spend hours at the bench for it. So, um, you know, yeah.
So I'm gonna pop in a question from YouTube. I'm not 100% sure what the question is, but I think it's about a bias. So it says, why is there a code bias?
Why is there a
Sorry, why is there a code bias?
code bias? I'm not sure what that means.
I'm thinking maybe like, is it about how, you have to do stuff on the computer? I don't know, in this area.
Maybe it's about
I
some
think
of
it's
our
about codon bias.
bias.
Oh, okay, sorry. Yes, that.
Why is there codon bias? So there are lots of ideas from a long time actually about why codon bias exists. So just for people who are not familiar with this, codon bias is the observed pattern in most genomes that we know of, where some codons are used to encode a particular amino acid more often than others, and the identity of this preferred codon seems to vary across organisms a lot, right? So people thought that okay, this was one of the first observations that came out of early days of sequencing genomes and genes, and people suggested that maybe some of this has to do with biases and mutations in themselves. So let's imagine there's a bias towards more, C to G mutations or C to T mutations. Then, by definition, you will get more codons that, use those bases, right? So there's certainly a role that the mutation biases seem to play in which codons are used more often and which organisms use which codons more often. There's also a role for selection, natural selection, and it's clear that for the large part, most of the codon bias that we see in organisms is probably explained by very weak selection. That is, very little bit of selection, not a whole bunch of selection. But there are also a lot of very interesting cases known where there is very strong selection. What I mean by that is, if you change the codons in some positions, there are huge impacts on the organism's fitness. So this would sort of mean that this would be under super strong selection. And so there are very well known instances of this as well. Also in the clinical context, interestingly, right? So there are mutations that are synonymous, which would change the codon but not change the amino acid, right? And these mutations are known to be very strongly associated with a number of human diseases or a number of things like resistance to specific kinds of antibiotics or drugs that we might take for a particular disorder or disease. So there's actually a lot of work that is now rapidly being done on trying to understand why these codon changes matter, right? And so we also did some work on this, and we continue to work on this to some extent. So what we found is that if you change some of the codons in this bacterium called Methylobacterium, where you make one version of a gene, a particular gene which is used very, very frequently by this organism and which is very important for it. Okay, it makes an enzyme, and that enzyme converts methanol formaldehyde, which is produced when they eat methanol, and that formaldehyde is toxic for cells, and so this enzyme it degrades that formaldehyde, right? It converts into something that is not toxic, and so we found that this enzyme now, if I change the codons that I use in this enzyme to in this gene that encodes the enzyme, if we change them all to the most preferred codons, okay, across the genome, let's say AAG is used much more than AAC. I change all the AAGs in this gene, so all the amino acids into AAG, without changing the protein sequence. We find that actually its fitness is really low. So this organism is almost dead, and it's correlated with its inability to produce this enzyme a lot. So this was kind of a bit of a shock, because then if this codon, putting in this codon everywhere, makes this enzyme non-functional, it's not producing enough enzyme, it's not expressed very much, why is that codon used so much, so much more in this organism's genome? And so what we found is it has to do with modulating expression of that particular protein. There's been a lot of other work by other people on other different enzymes, different bacteria, different organisms. And the funny thing is there's no single pattern that stands out. So we still don't actually know why changing some codons in some particular places is problematic or is really good in some cases, right? And so we don't really have a very solid handle on this. All I can tell you is it's complicated, which I know is not a great answer, but that's what it is. And there are many, many different reasons depending on where those codons are and what protein is it and how much is it expressed and who else is next to it and which organism. So all of these things seem to matter. Okay. And so we know some things. We know things. We know a few sort of patterns that are clear. So codon used at the beginning of a gene seems to matter a lot more. Okay, compared to codon used at the rest of the gene. In highly expressed genes, codon use seems to matter a lot more. In poorly expressed genes or lowly expressed genes, it doesn't seem to make that big of a difference. So we do see some such patterns that are clear. But there are many, many mechanistic reasons that seem to be playing a role. And so we don't quite understand enough to be able to predict what is going to happen for any random organism, for any random gene. We don't know enough yet. That's a long answer to your question, but anyway.
Yeah, so since it was on YouTube, it was a bit confusing. But yeah, coming back to the questions here, there are a couple more about, your one is about your research, and the other one is a detailed one. Maybe you can read through that from Srinidhi. So yeah,
Sorry, should I?
which one would you like to answer first? Go ahead.
Oh, maybe I'll attempt Srinidhi's question a bit. Let me try and understand it. So the question is, what are your thoughts on genetic determinism? Are we really in a place where we can say that one gene has one effect, sort of what is looked at in the CRISPR paradigm, where there is an attempt to tweak genes to tweak certain phenotypes? Um, so the as a concept, genetic determinism is, I mean, there is no one rule in biology in general, right, at all. So there are certainly some phenotypes which are controlled by single genes and some mutations which are very important, what we call big effect mutations, would have a very big impact on the phenotype. And so in a situation like the use of CRISPR to edit certain kinds of genes in certain ways, it would be possible to predict, okay, if I make this mutation, I'm going to get this phenotypic change. But by and large, what we are finding now from the last few decades of genetic work is that most phenotypes seem to be affected by many different genes and many different ways. And so, depending also on what kind of phenotype one is trying to alter or to influence, the answer will change, right? So many of the phenotypes, like famously human height or flower color and so on. Well, there are some cases of flower color which are, controlled by very few genes which you can manipulate, and there are no mutations that do that. But many, many traits that we might be interested in are actually sort of higher level traits in the sense they are the trait is a result of many functional and mechanistic differences in the organism, right? So there are many ways basically to change someone's height, and so it becomes more difficult then to predict exactly which mutations would do that. So in that sense, I think what is clear is the picture is very, very complicated. It's not usually going to be a simple, straightforward. Okay, I'm going to go in and make these tweaks, and then that's going to solve the problem. Uh, so yeah, so it's not that easy. Um, what is clear, however, is there is again the degree of heritability, which means um, how accurately or how reliably are phenotypes inherited from parents to offspring. We do know heritabilities of some of the traits that we care about. And some of them which are very simple Mendelian traits, of course, you've learned about them in your textbooks and so on. You're aware of them. But again, there are many, many other traits which we might care about, which don't have a simple Mendelian basis. And so it's going to be very hard to, twist them to suit our needs. So yeah, so that's that. Um, okay, maybe I will take a slightly different question now. Pavitra, do you want to continue moderating, or should I? Should
So
I?
you can go ahead with the one that asks, about the discoveries that are kind of exciting from the lab, right? Maybe you can.
Uh, yeah, sure. So let's see. So I told you about this work on codon bias, which has been really exciting and fun. And what was really fun, just as a quick follow-up for this work, so we showed initially that if I change codons that exist in the in this organism, we basically everything away from the wild type is a problem. Okay, to different degrees. So then we wanted to ask, well, how is that problem fixed? Because we hope that that would tell us what created the problem in the first place. So we allowed these organisms to evolve in the lab. And what we find is that they evolved really quickly. And in many cases, that evolution, that adaptation to the problem that we presented, that we created in the bug, was solved by single synonymous codon changes again, right? And so this was really exciting because uh, before that, people had hypothesized that codon changes would be under positive selection, would be favored in some cases, would be problematic. But here was a system where we showed that they were hugely problematic. And the solution also can be a synonymous change, a codon change, right? So that was not very well known. And what was also fun was that this occurred again and again. So in multiple replicate populations, the same thing happened. The same codon showed up, very, very strong effect, very large fitness effect where, the strain was barely able to survive. And then it was, doing almost as good as the wild type. So that was exciting. More recently, I'm really excited about a lot of the flower beetle work. So these projects were sort of longer term projects that, take a lot of time to develop. And so now they're ripening. So we have about 5 or 6 years of evolution that has happened in these flower beetles. So flower beetles will typically eat wheat. They're really good at eating wheat flour and they thrive on it. As some of you might know, if you've had old wheat flour in your house, it will almost certainly be infested by this pest. But if you give it ragi, you give it jawar, you give it corn, some other flours, it doesn't do very well on those flours. It sort of struggles to survive, struggles to reproduce. And so we wanted to ask, how does it adapt? Because it is, after all, a generalist pest. So it does manage to infest many different cereals and flowers, but we wanted to ask, how does it adapt to these different resources and habitats? So we had to evolve these different populations on these different flowers. And now they have adapted to these flowers, many populations, and we had some different permutations of different factors that we were curious about. And now that they've evolved, we are in a position to ask, well, what genes were involved in the evolution? What phenotypes were involved in the evolution? Which of the ancestral trait values really mattered most? How repeated or how similar are these trajectories of adaptation that we observe? What is the degree of adaptation? So these are all the questions that we're asking now. We're also asking whether their behavior has changed. Are they now more interested in eating or exploring these different resources? Those are really fun questions where you've seen evolution happen in front of your eyes, and now you can explore it much more. So that's a whole set of projects, and a bunch of PhD students in the lab are involved in this work. Another really exciting project that a piece of which we've recently finished, and this kind of developed during the pandemic, with a former student, Mridula, who is now doing a postdoc. So we found that so basically, I'm not going to the details of this project, but the point is to try and understand. Remember at the beginning, I said for codon bias, it was important to understand mutational biases. So one hypothesis is that codon bias comes up because, let's say, one particular bacterium is more likely to have transitions or transversions, right? And those may be more likely to lead to certain codons and so on. So we wanted to understand this very, very broad and basic question: why do mutation biases arise? How do they evolve? Are they beneficial? Are they deleterious to have that kind of bias at the genome-wide level? And why? So we collected some data to address this. So we made a bunch of new mutations and single mutations. We did a lot of sequencing to understand that, confirm these are single mutations. And then asked, okay, how much do they matter for the organism in terms of its growth rate? And we find that there are actually many, many more beneficial mutations than we imagine. So you think E. coli is very well adapted to glucose environment already, right? Or if some of you work with bacteria, you might know that we often culture E. coli in the lab on LB agar, which is a very rich medium, right? It's very luxurious, comfort, everything is there. So you think that this organism, which is, in its natural niche, is thought to live in our gut, at least for part of its life cycle, should be very happy and comfortable in LB. It is. And so you think that, okay, well, how can it get any better? It can't, because it's already really good at this, right? In fact, it turns out it can get better. And, as many as half of all mutations that we could explore in some environments turn out to be potentially beneficial. Now, the benefit may be very small, but it was nonetheless detectable in our experiments, right? So this was a bit of a surprise, and we're still sort of working with this. But using these data, we were then also able to ask, if we now shift the mutation bias, right? So certain kinds of mutations that they're more likely to happen, what is the impact on the organism? What can we predict is going to happen to adaptation? So we collaborated with a mathematician. We did a lot of work for this. Another former student, Gaurav, was involved. He mapped mutation bias in the phylogeny of bacteria. And so the upshot of this is I'm super excited about this work because we now have some answers and some predictions for how these kinds of mutation biases might evolve, the fact that they are actually important for selection, under what conditions will they be important. So this work shows here is how they will evolve, shows data that is consistent with these predictions, but it also makes a lot of new predictions. And so I'm really excited in the next few years to test these predictions, right? To understand this very fundamental property of all organisms, that they are biased in the mutations that they sample. And that sampling bias, that sampling effect, is a very major part of evolutionary theory that has been kind of ignored a little bit. Uh, yeah, so that's these are some of the things that I'm excited about from the lab.
Yeah, that sounds good. And for anybody who wants to know more and hasn't been following what's happening on, the Lab and Focus Twitter, please go and visit that. It's in the chat. So that's my quick plug for that. And let's move on to the next question. So, what were the core values that you established early on for your lab to have a healthy environment? So,
That's a good question, and I must say that take what I say with a little bit of a pinch of salt because our memories are famously terrible at being absolutely factual. So I will say what I thought was important and what I some things that I think are much more important than I realized earlier. So I think some of the core values that were very important for me was that people were nice and kind to each other, and people had a cooperative spirit, because I'm very much against unnecessary and excess competition within a lab. The whole idea of being a lab is a unit that functions to help each other and to work together towards common goals. That was a very, very important thing for me from the beginning. And that's something that I emphasized. And again, I was very lucky that that was the case in the people who joined the lab early on as well. Another core value um, which is very much a core value now, but I think was not as heavily emphasized early on, is honesty in all forms. Honesty about data, about mistakes, which is everybody makes mistakes. How to deal with those mistakes, and integrity in research, of course, is something that is discussed a lot. But I think it really is important in everyday life in the lab, and to emphasize that from the beginning and be willing to deal with the consequences of it, right? As a lab, as a faculty, is something that takes a little bit of practice and learning and figuring out how to deal with it. I think another core value is hard work, but that's not unique to my lab, in any way. Um, but I think from early on, I was very clear that people have flexibility about how they work, right? And again, that value has always been there. That it's important that people work hard and people make effort to improve. And everybody who comes in will need to improve on something, and some things they will already be really good at. But I think what I have added more recently is the idea that everybody starts from a different place, and that's perfectly fine. And what's really important is that the starting is at some level which is high enough that it's going to go somewhere, but also that the improvement is what matters, right? Because everybody will come in with different sets of expertise, different skills, different abilities, and that is fine. But everybody must be striving towards getting better as a scientist overall, and that's important. And what that means will be different for each person. And so what that also means is I have to learn what that person needs or wants out of their career, out of their time in the lab, and how best can I help that person to get there, right? And sometimes there's been a breakdown in this whole process. Sometimes there's been a breakdown in communication. Sometimes there's been a breakdown in um, you know, the students' understanding of what they want, sometimes my understanding of what they want, sometimes my understanding of what I want or expect from a student. And so I would say that has been one of the biggest learning curves for me, or important learnings for me from this whole process is how to communicate this clearly and make sure that these breakdowns are minimized. They will still happen once in a while, or it cannot be helped, but at least I need to make an effort and be clear about this. So that's I take that very, very seriously now, much more than I used to earlier. Thanks for that question. That's a very good one.
Great. Yeah. So I think, that's something for anybody who wants to go into either running their own lab or joining research also needs to think about, right? And so I have a question now. We've discussed kind of like the daily life in the lab and everything during this week, right? But let's say pre-COVID, I mean, I've been seeing a lot of trips, like your field trips as a lab. Did those happen often for data collection, or was it rarer?
Oh, so my lab now no longer has projects that really go on in the field. So we have had no reason to do field trips this year. So the last project we did, which required some field work, was working with butterflies to try and ask what their microbes, associated microbes, might be doing. And for that, we needed to collect some butterflies from outside. And so that project has now sort of more or less come to an end. There's a little bit of an interesting side project that's left to put together. Um, so that will happen soon. But yeah, the work has stopped. So field work is no longer sort of something that the lab does. Um, Unfortunately, I enjoyed a lot, but we have a lot of projects that are also exciting in the lab, and so that you can control things much better. And yeah, variables are in your, you know, you can understand them much better. So I think we're going to do that for the near future. So COVID didn't affect us too much in that sense. The ability to do field work was not so important during this period for us.
Okay, great. Yeah. And I guess, I mean, a lot of ecologists did manage to go back to the field anyway, so that was good. So I think right now nobody has got any new questions. Yeah, sorry. Okay, there's a question about. I mean, I think this might be outside your wheelhouse, but someone wants to know an interesting and important fact about our immune systems.
About our immune system, yeah, sorry, this is, I don't understand. I don't work on the human immune system at all.
Yeah.
And on the projects that we had that dealt with immune system of insects, they were all sort of driven and led by a fantastic postdoctoral fellow in the lab in Rose, and that was really his area. And so I learned a lot of immune function about insects, insect immune function, because he brought in these projects, and it was fun to learn. So I can tell you a little bit perhaps about insect immunity, but you really should talk to Rose for this. Um, yeah.
Okay, sure. There's another question about being a young female faculty in your field. What would you change for a newer generation?
There are many parts to this question. Maybe I'll address the part which just has to do with a new faculty in my field, because my field is very poorly represented in India in general. So even though I sort of fall in this broad category of ecology and evolution, a large part of my work and the lab's work has focused on molecular evolution. And there are very few people in India who work on molecular evolution. What is molecular evolution? The field tries to understand how molecules evolve, and to use molecules to understand the evolutionary process. The second part is done to some extent in India, of course. Like, people use genetic data to infer things about populations, about wild populations, about, in conservation or so on. That is there. But that is not making conceptual advances in the use of those tools either, right? Typically. So there is very little representation of that field in India. And the first part, where people are trying to understand the evolution of molecules itself, is extremely underrepresented in India. So that just means that it's very hard for people in my lab to have a peer group to talk to, or for me to have colleagues to talk to. So um, it's been largely the case that I have to talk to colleagues outside to get any feedback, and to really have a good conversation about that work. which is also fine. But I would really wish that there were more people interested in molecular evolution and doing sort of primary work on molecular evolution in India. Um, Apart from that, I think in general, like I was saying earlier, although there's a lot of work on conservation ecology in India, um, again, sort of very, very basic ecological research is not a really big deal in India and is not pursued a lot in India. Similarly, with evolutionary biology, there are very few evolutionary biologists in India in general. And so all of these have been challenges because I am somebody who really likes having a community to talk to listen to. I really enjoy conferences and meetings where, I get to listen and hear about what people are finding out. And so since I enjoy ecology and evolution talks a lot, it's a bit difficult to get that fixed. So I miss that, and it has been difficult for me to deal with that. I tried to set up some of these meetings on my own within India, but they didn't sort of take off very well. And the reasons are many and complicated, so we won't go into that at the moment. But um, but that has been fun. Uh, about being a young female faculty, I think I've lost the question. Hang on. Right. Where's the question?
Sorry, it went to the answer tab.
Yeah. Young female faculty, right. I think for my field specifically, I don't think there's any particular problems of being young female faculty or whatever. I mean, in fact, I think in general, ecologists and evolutionary biologists tend to be quite open, quite uh, aware of issues of gender and so on. And I have not faced any troubles in this regard at all. I don't know of any major troubles that any colleagues have faced in this regard as well, beyond sort of the problems that we know exist in India in general in our society about being a young female, period, right? This is a problem in India and this is difficult in India. So I don't think it's specific to the field that I'm in at all. In general, what problems have I encountered as a young female faculty? Also, very few in the big scheme of things. Okay, there are a lot of everyday irritations, like daily stuff. And again, this is not to diminish either things on the spectrum. I'm lucky that at least where I work in general, I don't face many of these problems at all. But I know for a fact that lots of people, lots of colleagues in India who are young and female and faculty have faced a lot of trouble. So those are realities of everyday life and professional life as well. And I think some of the most common ones, um, which might seem very small but add up to be really, really frustrating and take a toll on you over time, are the ones where you don't get recognized, the same way. You don't get listened to the same way as male colleagues do, as older colleagues do. Um, and that feeling that you don't have a voice, even if you are expressing your thoughts and opinions, is what is most frustrating. And this will change. And I have already seen some change in the few years that I've been here. But it requires two kinds of things. Um, One is a top down kind of pressure, right? Where there have to be strong rules and laws and strong statements or strong action from, heads of institutes, from government, from authorities of some shape or form, to say that this is acceptable behavior and this is not acceptable behavior, and to have clear repercussions for bad behavior. The second, however, which I think is much more important in some sense, because that is what brings attitudinal shifts, and which is something which is in the control of all of us, right? At the small level, at the grassroots level, is the change in attitude that has to come about. And that means that each of us has to every day talk to people around us, especially as faculty, in my role as faculty. I must make sure that everybody in my lab is aware of what is bad behavior and what is not tolerated, right? And this applies equally to men and women in the lab. Um, so, so I think those kinds of small changes have to be made. I mean, so commonly I get emails that start with "Dear Sir," and some of them are just plain irritating. Like, did you not take a moment to look at my website? Uh, where I clearly say, by the way, don't write to me without looking at artwork in the website because it's a waste. Why write to me saying, I want to work on mass spec or proteomics? We don't do that stuff, right? So if you've done that, clearly you can see that I'm female. So why write dear sir? Administrative people often also do that. And so at some point I wrote this email to my institute and to everybody saying, please stop doing this as a default. And I would like to think that there has been some change due to that. And maybe this has to be done repeatedly. We'll see. But these are very small things that all of us can do in our daily life. Small things that you observe somebody doing. Go up to them and tell them, don't do this. This is the reason why it's important. So hopefully that will lead to changes in attitudes and sort of small shifts in people's thinking, which I think must happen because sort of top-down laws and rules alone will not do that, will not have the impact that we need to have. So we need both of these processes working. Um, yeah.
Yeah, and I think that your answer also applies to the next question we had, about how science institutions could address these issues of diversity. But maybe you want to add a little bit more about that, and also, issues of diversity across gender and also class in Indian science. You mentioned that you might not have faced outright discrimination, but what about maybe like a lack of representation, as you mentioned?
Mm-hmm. Yeah. Um, I think the answer is fundamentally similar, to what I just said, that you need to have pressure from the top, pressure from, like a carrot and a stick. So you need a stick which is looming in front of people because some people will not change unless there is a stick. But we need the carrots, but we need the sort of everyday small shifts in thinking. And I think a lot of that comes down to exposure. All of us grow up with certain things that we see around us, and they become normal for us, right? Because we simply don't have the capacity to think about every single tiny decision and thought that we have. So a lot of stuff that we take for granted just comes naturally to us. Is partly at least born out of the environment in which we grew up. So as adults, if there has to be a change in our thinking and our behavior, it's going to be hard to change it. And I think the best way to do it is to have very good examples around of how things can be better, right? And so that is again something that is in our hands. Um, and those of us who are in a good position, where we are lucky to have a lot of resources, a lot of, um, I don't want to say power necessarily, but I do want to say power in some ways because I do have, for example, I do have the power to change or decide how my lab functions, right? So that is a tiny bit of power, not a whole lot of power. But within that, it's up to me whether I can use it very well to make sure that people who go out of my lab um, have been in an environment where they are exposed to issues of diversity, to be able to think about it, to see how you can function without having barriers um, show up, and hopefully then that becomes their normal. And then when they go somewhere else for the next part of their career or a different career, they take that normal sense of normalcy with them, and then they can make change wherever they go if it's needed, right? So I think we need to lead by these small examples in our own small circles, at least, at the very least. And the law will, catch up at some point, or the law does do some things already, but our attitude has to catch up with that in some sense as well. Um, yeah, but I mean, this is a complex question and a complex problem, and I can't claim to have all the answers. But it seems to me that this is where I would start, at least thinking about it.
Okay. Yes, thank you. I mean, it's obviously a complicated question, as you said. So, coming to a long kind of question about Darwinian principles of competition and struggle for existence. So, do you think that those have gotten more attention than they should have? Um, and then they ask, now that we know about Wood Wide Web, the existence of fungal networks in forests within species and between species, can we say that biologists have ignored the existence of cooperation compared to competition?
No, I don't think so actually, because even with cooperation, there is still a fundamental competition for limiting resources, right? And in some sense, you can think of cooperation as one strategy to overcome the shortage of resources. Um, and there has been a lot of work on cooperation and cooperating individuals and populations in the wild. Of course, we are understanding these kinds of processes where it involves microbes a lot more in the last few years because of the methods that we didn't have earlier, right? So things like the world, Wood Wide Web and so on, where it deals with mycorrhizal fungi who are underground and who are connecting lots of different plant roots and so on. We simply didn't have the tools to be able to understand and track exactly what goes on in those networks until recently. So the extent of that, perhaps, of a single network and its importance, we may not have known. But but clearly, cooperation was a very big feature of evolutionary theory from the beginning. Um, and the classical example is, of course, social insects, right? And the idea that they are seemingly altruistic. And how does this altruism evolve? How does it maintained? There are giant societies of social insects, uh, you know, all over the earth. How do they maintain these structures without seeming competition? And the answer is that they do face a lot of competition. In fact, so it's not that competition doesn't exist in these societies. It is there. It is there at every level. Um, and they've just found a way to work with it using cooperation. So I wouldn't say necessarily that it's been ignored. Uh, I mean, different things are in fashion in different points of time, in any field of inquiry. And so we've gone through those kinds of ups and downs, and fads come, and something is hot one day, it's not the other day. But I think there has been a lot of research on all of these problems uh, over time.
Okay, great. So I hope that helps. Maybe you can go and look up some of the early theories. I mean, not you, Deepa. I meant the person who asked the question. So there's a question about contamination in your lab. Working with insects, how do you maintain them separately and make sure there's no kind of contamination?
I'm not entirely sure what contamination you're referring to, but maybe I'll try and address it in a few ways. So we don't want these pest insects to get out and yeah, get out everywhere in people's homes. So we do have this work happening in a different closed room in our floor. So that's sort of preventing our pests from going out. And we freeze everything that comes out of that room, use flour and so on, so on. So we kill everything by freezing it before it gets dumped in the waste. So that's one set of contamination. We, of course, don't want other insects to, go into our uh, stocks and our populations. And so we do the same thing with bags of flour, which we buy. We buy lots of flour. Yeah, every week. And so all of them also get frozen before we use them. And that means that we are able to kill, any other insects that may be already infesting the flour. It also helps control fungal contamination to some extent. For other sort of microbial contamination like fungal mold contamination that might happen in our stocks themselves, we have some ways of managing the population such that we minimize those molds from spreading around. And so that's pretty standard stuff in the lab now. So yeah, that's the different kind of contamination we would worry about.
Okay, awesome. I think we'll wrap up here with just one last question. Since everybody has heard how awesome your lab is, uh, I've directed them to look at your join us page on the adaptation lab.insight. But also, if you want to add anything, are you looking for anybody at a particular level? I think you're looking for interns.
Interns, actually, no, we have a lot of interns at this point,
Okay.
more than we can take. I am looking to hire a graduate student maybe at some point. Um, but I'm especially looking for a postdoctoral level person, particularly somebody who might be interested in working on microbial evolution and who has some experience with that kind of work. I would especially like somebody who has some experience with evolutionary theory or evolutionary biology in general, because I think that's important um, for this person to really be able to function at the level I'm thinking about. The trick, of course, is funding. And so I'd be happy to help the person write grants and fellowships to get funding because I don't have a funded position at the moment. I'm hoping to have one free, maybe sometime next year or so, but we'll have to see that. So in general, I'm interested in, hiring a PhD student or a project assistant or a postdoctoral fellow, but yeah, there are some constraints of funding. So, but write to me and we can discuss what's possible.
Okay, great. So, Deepa, I just want to say thank you so much. And thank you to everybody who asked questions. And obviously, please write to Deepa if you're really interested in tackling one of the projects that she's also discussed today and future ones that you might have in mind. And I think we'll wrap up here. Deepa, would you like to add anything for people who are interested in this area, for instance, maybe
Yeah,
young students?
yeah, this is exciting. Evolution is really exciting, and I hope that some of you make some effort to read, popular science articles or books about work in evolutionary biology. Especially in India, there's so much to do because there's so many interesting organisms around us, and we have no idea how they evolved, how they behave. Um, and so there's a lot of questions that any of us can answer, and we don't need high level of techniques to address some of these basic questions. There's a lot to be done. There's a lot of new things to be discovered, and evolution is really fun. So I hope you take time to read and think about it a bit more and get excited. And if you have any major questions, do write to me or write to people in my lab, and we'll be happy to, discuss with you more. Um, yeah,
Okay,
thanks a lot.
thank you. Bye.